Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. Today we will talk about the photosynthesis, that is the overview as well as the explanation. So photosynthesis is very important for the plant. So here is the plant which that contain a shoot system as well as root system. That's a root system is non-photosynthetic, that's not chlorophyll are present in the root, so that is why not so photosynthetic. It's not used for the absorption of light. On the other hand, if we will talk about the leaves and stem, that is able to photosynthesized mechanism due to the their organ is composed of the chlorophyll pigment. So here is the cell which that is I am drawing, contain nucleus, endoplasmic reticulum, and here is the green color which they are showing the plasma chloroplast. This chloroplast organelle is very important for light absorption, and that is the used for the light reaction and dark reaction in the form of chemiosmotic ATP production as well as NADH production, and that's dark reaction will be in the form of Calvin cycle. So here is the thylakoid membrane, which that is basically thylakoid membrane contain granum, and each granum is collectively called grana, and that's a stroma are present in the fluid-like structure are scattered around the granum. So is the outer and inner membrane which that is present in the form of chloroplast. So here I am saying that is the granum. So one granum I pick out, and there's a lumen, contain lumen inside, and outer is the stroma surrounded the thylakoid membrane. So that is basically here is the structure which that is outer membrane of the granum, which that is looking like here, and that is green color which that is showing chlorophyll pigment as well as white color is the carotene and chlorophyll A, chlorophyll B, and different type of xanthophyll, fucoxanthin, that is a different pigment are present. So we will discuss about the just chlorophyll. So chlorophyll contain a ring and tail structure. The ring is known as porphyry ring, and phytol tail is the hydrocarbon containing phytol tail. That is the ring is used for the absorption of light pigment. So the two chlorophyll, chlorophyll A, chlorophyll B, the difference is here. There's the 72 hydrogen in the form of chlorophyll A, and in the form of B is the 70 hydrogen, and oxygen 5 to 6 difference, and that is the light absorbing pigment which absorb the light for light energy conversion into chemical energy. So that's easy to understand about these things. So next we will talk about that is overview about the photosynthesis. So here is the light which that is come into the membrane of the granum, and that's ATP and NADPH produce. Why? Because photolysis, hydrogen physically decompose into our lies due to the oxygen production. So oxygen we release, and here is ATP and NADPH produced by chemiosmotic synthesis of ATP, and that's going to the Calvin cycle to produce sugar as well as fat and protein. Now here I am discussing about the bacteria which that is also photosynthetic, but the difference is about the photosynthetic bacteria as well as the plant is a huge difference due to the sulfur instead of the water. So remember about that. Bacteria contain sulfur, so here is the chemical equation is equal to the chemical equation of the plant but oxygen instead of sulfur. So that is the difference. You can stop the video and identify it. Next we will discuss about the in the form of graph. So we can say that the basically here first of all we will discuss about the electron transport chain in the sense of chemiosmotic synthesis of ATP as well as NADPH in the case of light reaction. So here is the sun rays will be scattered and um, fall on the photosystem 2, and this photosystem 2 is basically water. Photolysis is known as hydrolysis. So that is basically water will be split into oxygen. One water molecule will be converted into the one our two oxygen and two proton as well as two electron. So here you should remember about that the plastoquinine, which that is the protein which bind with the hydrogen ion, and two hydrogen ion bind by a two proton to two electron and proton. So these electron basically these electron will be go into the um, uh, when proton go down into the lumen here, and electron will be further enter into the cytochrome BEF, that is cytochrome, and that's the plastocyanin will be accepted, and this plastocyanin will be further through the into the photosystem 1. During the photosystem 1, that's energy is too much low, and there's a absorption of photosystem 2 contain 700 nanometer and 680 nanometer absorption of light in the photosystem uh, 2, photosystem 1, sorry. So here is the NADP H reductase enzyme, NADP plus reductase enzyme, which they are used for the exception of proton, and two basically two electron and two proton will bind by NADP plus to NADP H conversion. So these thing is the plastocyanin will go into the ferredoxine, and ferredoxine accept to go into the NADP plus reductase enzyme, and this NADP plus reductase enzyme will be make a NADPH. So now we will discuss about the NADPH production is done. Now we will discuss about the ATP production. So the proton which that is come inside in the lumen from stroma during the electron transport chain, further flowing of the electron transport chain. So these proton gradient will be produced, and that's ultimately proton will be go outside for maintenance of the proton gradient to produce ATP from ADP. So that is known as ATP synthase enzyme. So now we will discuss about the photo excitation fundamental principle of light absorption called the Stack-Einstein law. In basically in this law, any molecules can absorb on one photon at a time, and this photon cause excitation of only one electron when molecule absorb electron. So one electron basically, if one electron absorb, there's a one proton is the basically one photon is required. Now here I am drawing the graph which that the 400 nanometer to 800 nanometer is the basically the range. So the maximum light absorbed in the different carotene is maximum 500 to 540 as well as 400 for chlorophyll A, and on the other hand carotene is showing in the graph which that is showing that the more light absorption. So here I am discussing about the wavelength of the plant, that is the wavelength of the plant downside, and the upper side is the photosynthesis. So the photosynthesis is more in the wavelength of 700 nanometer, but the lower here is also 400 because blue and red light is responsible. 700 contain red light, and maximum in the blue light is also as well. So the minimum is due to the going to the green pigment. So that is the basically the graph for the wavelength of the blue and red is more favorable. But on the other hand, if we will talk about that, you can pause the video and watch the graph, for example. So here I am discussing about that the photolysis of water. So here is the water which that attached by the plastoquinine to produce in the photosystem 2 oxygen and PKOH2, and this PKOH2 plastoquinine and dihydrogen ion, which that is iron sulfur protein, iron sulfur complex to cytochrome complex, and that's a plastocyanine, which that is accept, and plastocyanine will further plasto photosystem 1 to through the NADP plus, and this is the major reason for the ATP synthetase enzyme used for the hydrogen ion. And on the other hand, the electron which used for the making NADPH, and this NADPH is used for the Calvin cycle. So you should remember about that the Z-like structure is developed for the chemiosmotic chemiosmotic ATP production. So chemiosmotic synthesis of electron ADP to ATP. So synthesis of electron basically. So here is the light which that is scattering into the photosystem 2. Photosystem 2 is the 680 nanometer light absorption wavelength of the 680 nanometer. So basically here is that is the primary electron acceptor. So here is the primary electron acceptor which that accept the electron, and this electron will further throw into the membrane of the thylakoid. So thylakoid membrane which contain a pheophytin. This pheophytin accept the electron, and this energy is further drawn. There's a down the energy and low energy toward the plastoquinine. Why? Because low electron energy is acceptable for more further cytochrome BF complex. It's not go back. It's not irreversible. It's not reversible. It's reversible. So plastoquinine, which that is here, which there you can see, and this is a plastocyanine cytochrome BF complex, which that is. So here is during the downs going down the electron to produce ATP from ADP, and this electron again will fall into the photosystem 1, and this photosystem 1 is also required the light obviously, and this light when fall into the photosystem 1, that's light will be light is used. So here is the ferredoxine is used for the during the primary acceptor. Ferredoxine is used for the further proceeding of this process by the enzyme NADPH reductase, NADP plus reductase enzyme for using 2 electron and 2 proton to produce NADP plus reductase. NADPH. So this NADPH is used as well as ATP used for the Calvin cycle, and this Calvin cycle is the non-cyclic photophosphorylation. If we will talk about the cyclic photophosphorylation, so the here is the 2 electron which that again go into the primary electron acceptor to pheophytin. But why NADPH is not made? If too much NADPH is present in the form of for the requirement of the Calvin cycle, so it's no need to go for making NADPH, so it should go back toward the again cyclic activity from the primary acceptor toward the photosystem 2 side. So that is known as the cyclic photophosphorylation. That is due to the photo mean light, phosphorylation mean ATP production. So these now we will discuss about the Calvin cycle which that is required the ATP, and this ATP RUBP which that is ribulose bisphosphate or ribulose 1 5 bisphosphate conversion. These ribulose 5 bisphosphate which that is go into the by the fixation or carboxylation of the carbon dioxide, which that is enter into the to make a ribulose 1,5 bisphosphate into the 3-phosphoglycerate, and these step is due to the rubisco enzyme, and this is known as carboxylation. And 3-phosphoglycerate which will be used the ATP to produce ADP convert and produce 1,3-bisphosphoglycerate. It means 2-phosphate group. That's 2-phosphate group is basically 2-phosphate group is NADPH is used. This 2-phosphate group containing compound 3 carbon, that is the inorganic phosphate group will be released by the dehydrogenation, and there's a glyceraldehyde 3-phosphate will be produced, G3P, and this G3P is known as reduction, and this reduction go into the regeneration, and one molecule will be separate, and other molecule will go toward the RUBP by the different chemical mechanism. And this G3P glyceraldehyde 3-phosphate used for the making cereal crops, burger, that's all quality is due to the further proceeding of the these things is production. So that is 3-phosphoglycerate is produced by the fixation of carbon dioxide, and that's go by the use of ATP to produce 1,3-bisphosphoglycerate, and this used for the dehydrogenation to produce that things. So thanks for watching. Please subscribe my channel, and please comment below about that things.